Welcome everyone, myself Vaishnavi Vankar, student of BSc final year from Shri Shivaji Science College, Amravati. We are going to study on topic sterilization techniques under the guidance of Dr. Tushar Wankhede sir. These are the contents. What is sterilization? Method of sterilization techniques. In classification, there are two types, physical sterilization and chemical sterilization, then advantages and disadvantages, conclusion and reference. What is sterilization? Sterilization can be defined as any process that effectively kills or eliminates transmissible agents such as fungi, bacteria, virus, and prions from a surface equipment, food, medication, or biological culture media. Method of sterilization includes: first method, disinfection, destruction of all pathogens or organisms capable of producing infection but not necessarily spores. All organisms may not be killed, but the number is reduced to a level that is no longer harmful to health. Second is antiseptics, chemical disinfectants which can safely be applied to living tissues and are used to prevent infection by inhibiting the growth of microorganisms. Third is asepsis, technique by which the occurrence of infection into an unaffected tissue is prevented. In classification, in physical sterilization include heat sterilization, radiation sterilization, and filtration sterilization, and chemical sterilization include alcohol and aldehyde sterilization and gaseous sterilization. In physical sterilization, heat sterilization. Heat sterilization is the most widely used and reliable method of sterilization involving destruction of enzymes and other essential cell constituents. Dry heat 160 degree to 180 degree Celsius oxidative changes occur. Sterilization from thermostable products, moisture sensitive material, moist heat 121 degree to 134 degree Celsius. Hydrolysis and denaturation occurs. Sterilization is used for moisture resistant material. Dry heat sterilization, temperature ranges of 160 degree to 180 degree Celsius and exposure time up to 2 hours. Good penetrability and non-corrosive nature useful for sterilizing glassware and surgical instruments. Also used for sterilizing non-aqueous thermostable liquids and thermostable powders. Dry heat destroys bacterial endotoxin or pathogens applicable for sterilizing glass bottles which are to be filled aseptically. Dry heat sterilization applications are sterilization of dry powder, drug, suspension of drug in non-aqueous solvent, oil, fats, waxes, soap and hard paraffin, oily injection, implants, ophthalmic ointment and ointment bases, etc. In moist heat sterilization involves the use of steam in the ranges of 121 degree to 134 degree Celsius. Steam under pressure up to 15 lbs is used to generate high temperature needed for sterilization. Saturated steam acts as an effective sterilizing agent. Moist heat sterilization applications are sterilization for surgical dressing and instrument, containers, closure, medical device, culture, media, etc. Radiation sterilization, EMR, example gamma rays and UV light and particular radiation accelerated electrons. Major target for this radiation is microbial DNA. Use sterilization of heat and moisture sensitive products. Gamma rays from cobalt-60 are used to sterilize antibiotic, hormones, sutures, plastics and catheters, etc. UV light has low energy and poor penetrability. Surface sterilization of aseptic work areas for treatment of manufacturing grade water but is not suitable for sterilization of pharmaceutical dosage form. Filtration sterilization does not destroy but removes the microorganism used for clarification and sterilization of liquid and gases. The major mechanisms of filtration are sieving, absorption and trapping within the matrix of the filter material. Examples are HEPA filter used in the treatment of heat sensitive injection and ophthalmic solution, biological products and air and other gases for supplied to aseptic areas. Used in industry as part of the venting system on fermenters, centrifuges, autoclaves and freeze dryers. Membrane filters are used for sterility testing. Chemical sterilization. In chemical sterilization, firstly we can see in alcohol sterilization. Frequently used are ethyl alcohol and isopropyl alcohol for sterilization. This must be used at concentration 60 to 90%. Isopropyl alcohol is used in disinfection of clinical thermometer. Methyl alcohol is effective against fungal spore, treating cabinets and incubators. Methyl alcohol is also used toxic and inflammable aldehyde, formaldehyde, having bactericidal, sporicidal, and has lethal effect on viruses. Used to pressure anatomical specimen, destroying anthrax spores on hair and wool. In gaseous method of sterilization, the chemically reactive gases such as formaldehyde, methanol, and ethylene oxide possesses biocidal activity. Ethylene oxide is colorless, odorless, and flammable gas. The mechanism of antimicrobial action of the two gases is assumed to be through alkylation and sulfur-hydryl, ammonia hydroxyl, and carboxyl group on proteins and amino groups of nucleic acid. These are the advantages and disadvantages of sterilization techniques. For moist heat sterilization, advantages are commonly used and familiar to regulate, safe and effective, inexpensive, and penetration well. And disadvantages of moist heat sterilization are high temperature cannot be used for all atoms, not for atoms sensitive to moisture. And for dry heat sterilization, advantages are widely accepted as effective, good for moisture sensitive atoms, can dehydrogenate, inexpensive. And disadvantages for dry heat sterilization, few materials can withstand the heat, longer cycle time than moist heat. Ethylene oxide sterilization, advantages are for heat and moisture sensitive materials, inexpensive, can be used for electronic component, sporicidal. And for the disadvantages of ethylene oxide, require gas permeable packaging, long sterilization cycle, toxic, carcinogenic, and explosive, no standard cycle parameters. And for radiation sterilization, advantages are available commercially, no harmful emission, relatively low chemical reactivity. And disadvantages of radiation sterilization, expensive specialized equipment performed by outside contractor, degrades some plastic, rubber, and Teflon, and very dangerous. Vaporized hydrogen peroxide, advantages are useful for special application like isolators, inexpensive for heat sensitive materials. And disadvantages for vaporized hydrogen peroxide, can be corrosive to materials, respiratory irritant, no standard cycle parameters. Filtration sterilization, advantages are used for heat sensitive liquids, and disadvantages for filtration are release on good operator aseptic techniques to maintain sterility. These are the conclusion. Prevention is better than cure. The main way of control contamination is sterilization, which have been developed in the past few decades and currently in use in the industries for sterilizing medical instruments, foods, and pharmaceutical. Most of these methods are automated and efficient, environment friendly, have low running costs, can sterilizer on a large scale, highly effective against certain resistant microorganisms, and ensure complete sterility of products. These are the references. I hope you all understand the topic of sterilization technique. Thank you for watching my video.